Kelly is her name? Kelly described like the first most important thing I ever did in my life, which was help Aubrey degrade a SIMS project by finding some enzymes. And I'm going to tell you mostly about the next most important thing, or the most important thing that came after, which was helping Halcyon molecular get off the ground. And you've heard Ben talk about AI research, so grappling with complexity from a computational perspective. You heard Aubrey talk about cutting through all that complexity and doing something that really needs to get done. And we're taking the tools approach. We're taking the fundamental understanding approach of actually grappling with that horrendous graph that Aubrey showed, because we think we can accelerate progress in tools to actually maybe be able to grapple with that in a reasonable period of time. So we're on an epic quest, and if your eyes are really sensitive, so we're dreaming of this really bright future, and I've got some sunglasses you can borrow if it's too much for you. And we dream of this incredibly bright future, and we're gonna get there. I don't know if it's going to look anything like this, but this always makes me feel good when I look at it. So I work for a company called Halcyon Molecular, and I did something in the lab a couple of months ago, accidentally, by pulling out individual molecules that happened to look like the cover of Dark Side of the Moon. And my friend Mark Hamalainen went in and colored it like this. So those are individual molecules at the top, and then that's a mess at the bottom. And we're sequencing DNA by looking at individual atoms. So here's some individual atoms that someone else looked at. I'll show you some that we've looked at. And the whole point of this is how outrageously fast, cheap, accurate it's going to be when it scales. It's going to be the technology that's used to bring something to 7 billion people that will be a part of their medical record and will be a part of saving their life, and also a part of figuring out biology and grappling with that complexity. So the approach we're taking was, does anybody know who this is?
Richard Feynman.
So this is Richard Feynman, who in 1959 proposed how to sequence DNA. He was the first person to actually propose something specific about how to do it. He said use electron microscopes. And specifically, he was envisioning that the theories that had been worked out by this guy, Otto Scherzer, should be applied practically to electron microscopy. And then if you applied those principles based on the equation Scherzer had worked out, you'd be able to see individual atoms. You'd be able to actually just look directly at DNA. But unfortunately, nobody practically put Scherzer's theorems into practice until just recently in the 90s, even though he came up with them in the 30s and 40s. But there was a workaround, and that was adding heavy atoms to the DNA. So a ton of people worked on this approach in the 60s and 70s. And they managed to do things like this. They added heavy atoms to DNA, they stretched them out on ultra-thin supports. So they're throwing electrons through this, and they're capturing the ones that are more scattered by the proton density within the heavy atoms you see there. This was 1972. This was Michael Beer and Van Mudrynakis, who were the two main pioneers in the 60s. And then other pioneers in this work were Peter Ottensmeyer and Robert Whiting. They did about 20 years of research into this approach, and what set them off the trail, two main things, is that other sequencing approaches, biochemically based, started to work well enough to satisfy their curiosity. Their funding dried up, and there were fundamental issues they couldn't overcome. The main issue being that you can only see the heavy atoms. You can't see the backbone of DNA, the naturally occurring elements in DNA. So you get a lot of ambiguity in the order that you're trying to parse out from the single heavy atoms, which are the only thing you can see. So the problem as they identified it was, how do you actually correlate these heavy atoms, which you can see, to the positions of the bases that they're tagging? You can't see phosphorus, individual phosphorus atoms, or oxygen or nitrogen or any of the natural constituents. So you can't tell what's there. But if you can figure out how to get that consistently stretched onto these grids, you'd be able to determine the order of the bases efficiently. So this is the problem that our two founders heroically solved. Michael and William Andrick, Michael on the left, William on the right. They're here in this nice space station. And their solution was to actually take individual high molecular weight molecules and pull them into air, one at a time. Imagine a chain of atoms covalently linked. The backbone of single-stranded DNA, oxygen, phosphorus, oxygen, carbon, 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 repeating 1,000,000 times. Imagine that being stretched out into air in a chain. 1,000,000 of those, or 6 million of those, would give you a megabase piece of single-stranded DNA. They figured out how to individually manipulate molecules by using a piezo positioner and a specially coated probe. What you see here is a needle with a radius of curvature at the end of about 20 nanometers, and it's pulling out a mixture of many single strands that can be imaged optically. And the results of the protocol they developed have made some polymer scientists salivate. So here you see an arrangement of parallel molecules on a ultra-thin support grid, and this was early data, early proofs of concept. The gray stuff is a film that supports the molecules. It's about 5 to 10 nanometers thick. The big white bars are this much thicker supporting structure called a lacy film. And what you see at this sensitivity, at this resolution, are actually ropes of multiple molecules. And interdigitated between those are single molecules. You need those to sequence. This is a fluorescence version with 1 micron spacing between the strands, and a 3D rendering using something called an atomic force microscope, which is sort of a nanoscale stylus that goes in and measures sub-angstrom differences in height. So you see this in the middle there, you see a single strand, and these are ropes on either side. All of it sitting on a silicon surface. And this is the whole point of it, is being able to read out that sequence consistently. These are individual osmium atoms on a known template. This was one of the first nice pieces of sequence data we ever got. And those are individual osmium atoms. This was taken on the team 0.5 at Berkeley. And the sequence we know to be GT, GT, GT repeating. This one's about 70% true positives with regard to the underlying sequence. So Michael and William figured this out, and they did it without any formal training in science. They were dropouts, and I think this might inspire some people to say, "What am I doing in school? I just want to figure stuff out." So I think they figured out one of the most. They were the first two people on Earth to know that you could do this very fundamental thing with single molecules of DNA, and they did it without college degrees, which I think might say something. And the founding of our company was very much inspired by the big dreams that we're talking about here. So William was in the book section at Barnes & Noble one day when he thought about all the things he would never get to know if he didn't live long enough, and all the places he might never visit. So you'll hear him talk online about living long enough to see the stars. And they found me, and at the time I was at University of Arizona. I was struggling with the molecular circuitry of Drosophila, trying to figure out some events involved in how the wing is formed on a molecular level. And it was kind of a waste of time that I put 3.5 years into because of the tools. There's no point in wrestling with the complexity of this mess, from my perspective, especially since I was hunting down mutations, looking for molecular lesions, putting a lot of work into that. and I wanted better tools. The tools for sequencing were horrendous. So we came out to Silicon Valley. We met these guys, Peter Thiel on the left, Luke Nosek is there too, and we got funded to actually do this with a real lab, and got George Church, one of the world's leading genomics tech dev people, to join our scientific advisory board and come hang out in our ball pit. And also got Elon Musk, who you might have heard of. He's getting us all into space, leading the effort at SpaceX, and here he is flying a spaceship. And also got a tenured professor to walk away from a job for life to lead our microscopy division. This is Andrew Blaylock. And since then, we've seen more of the world at single atom sensitivity than all of the groups on the planet combined. And we also have the first synthetic chemistry project ever where looking at individual atoms and the molecules we make is becoming an analytical procedure. And this is some of our data giving you an idea, just conceptually, how far we've gone. It's not even to scale. It's just, here are single atoms on a carbon support. And it's just part of what we've mapped at the level of being able to see single atoms. So, something else I want to bring up, especially because I'm here, is the connection between Humanity Plus and Halcyon that I think none of you know, is that we founded a club at University of Arizona called the Hplus Club before we came out to Silicon Valley. And one of the reasons that WTA changed its name to Humanity Plus is that they like some of our branding. So you kind of took our name to some extent and we gave it away, but I don't think you took our unique approach to campus advertising. And I'd just like to show you some of the flyers that we put up on campus. So aging is bad for you. So a lot like smoking cigarettes just to keep aging. This brought a lot of people in. We had 30, 50 people at these meetings who had never been exposed to any of these ideas before. And this one also got a good response. But the one that the one I was actually a little bit worried about before we put it up all over campus, and I'm surprised we didn't get any like outraged letters to the Wildcat about this one, but it did get attention, which was the purpose. Getting the attention of the right people who felt the answer to that was clearly no. So why are we doing this? So you might have heard that the human genome was sequenced. It's never been sequenced. If you put everything together, the master reference human database is still missing millions and millions of pieces and has lots of gaps in it. It's because the technology isn't there to actually complete it. And we're still kind of running on the fumes of technologies that are not quite there yet. And those technologies look like they're still not going to be there at a cost, at a completeness that's going to actually make getting your genome sequenced a clinically valid proposition, something that's really going to make a difference for you. So, for those of you who don't realize it, we're all machines. We're nanobiotechnology that's been going for 4 billion years now. And sequencing, as reading off this information, it's fundamental to being able to hack it, to being able to learn how to program it. From the most fundamental perspective, we're doing something that will not only be upstream of medical relevance, of figuring out all the loci that contribute to diseases, but actually being able to turn all this machinery into the ability to make any materials we want cheaply. And not just that, but all the other things you could do once we can honestly say biology is an information science. We're not just making guesses when we give you a small molecule, but we have some precise sense that it's actually going to help you. And we want to get there as soon as possible. We want to make the best tools to get there as soon as possible. And that's all I have to say. I'm open to questions and open to going from those fuzzy outlines to what you're really interested in. Thanks.